been here chatting with Jessica for about 10 minutes now, and it's amazing how much we can just talk in 10 minutes. Imagine if we had more time. So, the topic of today is machine learning for genomics, and I find it really amazing because if you've been following Jessica's path, her first talk in English was actually here in GDG Cloud London, and she's been giving many more talks in here. And she's recently became a Google developers expert, and she's like, okay, now we really have to give one more talk at GDG Cloud London, and I couldn't be any more prouder of her for everything she's been achieving. And it's such a pleasure to have you here today. And let's see who are the people who are already here. We have Dimitri, Giacomo. So there's many people ready to see you give your talk. I just sent like the link to everyone in all the groups that I'm in WhatsApp. So hopefully more people are going to join you. And without further ado, I'll let you introduce yourself, Jessica.
Hi, everyone. How are you? And I like so much to be here again to talking about genomics, bioinformatics. First, I would like to explain because bioinformatics. Bioinformatics is a topic that I like so much, and I am enthusiast, and I'm preparing to my master degree in computer science, focus, and bioinformatics. I thought, I'm going to talk about genomics. People need to understand what's genomic. Today, we have a pandemic with COVID-19, and genomic is a topic in the world. Let's learn about, to understand how to work the science and how machine learning can help genomics and biology. Okay, let's begin. And recently, became a GDE in machine learning. I'm very happy because it's very important to me, and I can help many people in the world and the community to apply my knowledge to other people in machine learning and biology, because I like so much these two science. So, who I am? My name is Jéssica. I'm a software engineer and data analyst in Rio de Janeiro, Brazil. Recently, I became a machine learning GDE. Wow! I'm a bachelor degree in information system for Federal University of Sergipe. And today, I'm preparing to my master degree. I'm hoping this pandemic help me first, because selective process are complicated here. So let's start. Hello, everyone. I love so much cats. So in my presentation, always be happy. Be happy with cats. So let's talk about genomics. First, genomics is related to biology. Okay, but biology and computation, yes. We need to apply computation and many science. In my research, I like so much to apply computation, biology, and finances too. But in this presentation, I'm going to talk about genomics and biology. We need to understand some topics in biology to understand why apply computation genomic, and this explanation is very important to apply the better form. So genomics is a branch of molecular biology. Let's emphasize molecular biology. We need to microscope word, focus and study all aspects of genome or sequence of DNA. Genome is all these genes in the human and other organisms. Gene is a particular sequence of DNA. DNA is formed in 4 bases, and these bases form sequence. And a specific sequence are called gene because genes decodes proteins form humans, animals, and other organisms. And our dynamics are billions of bases, but we need 100,000 genes in humans. So other organisms is other quantities. The human genome is divided into 6 chromosomes in 23 pairs. So the last pairs is the sexual pairs to determine feminine and masculine. Genes are located in chromosomes, okay? Because we need to think, genes where? Where are genes in humans? Are located in chromosomes. In this photo, we need chromosomes of humans. In this case, it's a woman. We need 23 pairs. You can see it in this image. And the last pair is a sexual chromosomes. It's XY and XX. Okay. In this case, we need to understand that some situations we need more chromosomes. It's causes some syndromes, but it's not a topic here. But it's important to say about this situation. So some people can have more chromosomes. Depends on the situation. So this photo is my source in the Encyclopedia Britannica. After this explanation, let's understand the central dogma of molecular biology. So we need the DNA. DNA is an acid. The acid, ribonucleic acid. We have an RNA, a ribonucleic acid. And these interactions form proteins, form organisms. So let's understand this process now. We have a DNA. We have a process called replication. Replication is replicate DNA, but DNA passes information to RNA. This process is called transcription. DNA passes information for RNA. RNA copy this information and make proteins. This scenario it's called translation. RNA make proteins. Translation. RNA replicate the same molecule. It's replication. DNA passes information, decodes information for RNA. It's called transcription. RNA make proteins. This called translation. In some cases, some organisms, virus, we need a special type of virus called retrovirus. This virus don't have a DNA, only RNA. Its retrovirus make a special process called reverse transcription. It uses your cell, RNA yours, and make a process called transcription to transform RNA in DNA. through this enzyme called transcriptase. Okay, these organisms make this process only RNA. It's retrovirus, only virus. But and many organisms follow this process: DNA transcription, RNA translation, proteins. Animals, humans, plants, but some virus, especially retrovirus, HIV, it's a retrovirus, make a reverse transcription. Okay, so this process is important. Why? Because it's a process of life. Life follows this process and forms humans, organisms, animal, plants. These informations form an organisms. Your eyes, your hair, your hands. You are a code. You can understand DNA like a code, like a database. It's a code, and DNA are a code. Can determine your characteristics. I love so much the science because it's explain life. Life is very, very important. So DNA process, central dogma of biology. Let's understand now sequences, because and I say we are code, but code in DNA form sequence. We need to analyze this sequence because the sequence form genes. Genes form proteins. Proteins form lives. Proteins, it's a molecule very important in life. This form your muscles, your bones, your blood. Proteins determine several processes in your body. Disease, interaction between molecules of proteins determine diseases, cure. So this situation is important because understand how to work this process in the body, we can find cures for several diseases. In my case, I like so much to just research about disease and drug targets for disease. In the situation, we need to understand proteins. So, DNA sequence. I don't know, but many people have listened about genome project, human. This project is so important to science because she sequenced all the genes of humans, human genes. So, according to NIH, National Human Genome Research Institute, DNA sequence is a laboratory technique. So, it's a laboratory technique. These sequences are made in laboratory, used to determine the exact sequence of bases. It's to understand this sequence is not age, CEO. No. It's important and understand the sequence of bases. It's determine this life. So A, C, G, and D. Adenine, cytosine, guanine, and thymine. I don't know how to say in English very well the substance that form these bases. So in a DNA molecule, the DNA base sequence carries the information a cell needs to assemble protein and RNA molecules. Remember, DNA is deoxyribonucleic acid and RNA and ribonucleic acid. DNA sequence information is important to science investigate the functions of genes. Genes make proteins. These proteins form your body, your process. In our body, we have many processes. Okay, understand this process. It's important to find cures for several diseases and treatments too. For example, cancer. And exists many research about cancers, and genomic it's a target. But genomics it can be about hereditary or no, because many genetic processes happen in your lifestyle, because your lifestyle it's not hereditary. So cancers is related about your lifestyle, not only hereditary. So we discussed about genomics, but why apply machine learning in genomics? We can see in this analysis, these sequences, this DNA sequence forming many databases. When think about databases, many data, big data, so we need to analyze this data. But human analyze data, it's complicated, it's difficult. So computation, it's a good tool to work this information about life. Some patterns in the genes may indicate a predisposition to certain disease. An example about the situation in the gene BRCA about breast cancer. So exists many researches about BRCA genes to find the treatments, to find predisposition in some people. Machine learning can be useful to identify these patterns. In addition, my work is about this topic and specific, pharmacogenomics. Genomic data can be combined with clinical data. Clinical data is about patients. It's your symptoms or exams. So, to identify drug targets or how genes affect a person's response to drugs. It's a pharmacogenomics. It's related to pharmacology, drugs, etc. So we need disease, we need treatments, we need to identify drug targets and how these drug targets affect a person too. In genomics, we can combine many data. We combine genomic data with clinical data, historical data, when we can make many analyses. And with many data, many scientists in the world work with biology. So we have many data repository about biology science. We need the nucleotide and protein sequence. Proteins also form sequence too, but more bases. It's on 20 bases, I guess is. Nucleotide and protein sequence. Protein structure, yes. Proteins is a macromolecules, so it's big. We need to understand, we need to analyze this structure because the structure form different proteins. So it's important to analyze the structured protein too, because this protein structure determine some proteins specific. Genome, it's all the genes and the humans and all organisms. Genetic expression. Genetic expression is how genes make this protein. This process, how genes make proteins works, it's called genetic expression. And by bibliography about several studies about biological topics. And I put some repositories, but we need more. But we have more. NCBI on UniProt is specific to proteins and the structure proteins, etc. Ensemble. PubMed. PubMed is article and researches and specific. Biological topics, we're talking about biological topics, so now we're talking about machine learning. I like so much to discuss about what is machine learning because I understand that many people don't know what's machine learning. So let's talk about this. Machine learning is an application of artificial intelligence, AI. It's important to emphasize this topic. Artificial intelligence, it's big. We have other types, and machine learning is an own application that provides systems ability to automatically learn and improve from experience. IBM say the value of machine learning is that allow you to learn from that to predict the future. Not exactly the future, but in this case of the genomic, we work with sequence. Not exactly future, but the next. Next can be future or no, it's a sequence. And genomic, we work so much with sequence. In sequence, we need a special type of machine learning. We obey this process when we work with machine learning. The machine learning process and genomics, we work too, this form. Collect data. And biological topics and biological research, collected data is relevant, it's a very important process and phases, okay? We need to select data sets. Some repositories, these repositories can be very useful to extract data for your researches, for your applications. For example, we can combine these several databases. In my case, I'm going to combine an approach with Assembler. I'm deciding yet. I'm deciding how, which repository I will use in my research. Probably UniProt. Collect data. We need to collect data, understand what the significance of this data for your model. Select a data set with relevant data. Let's emphasize relevant data. We have to better learning. Significance data. It's very, very useful for you to achieve this better result. Prepare data. In genomics, we have many data that needs transform and feature engineer too. So feature engineer, I would like to emphasize, it's an important topic here. Feature engineer. Depends on our model. And prepare the data, manage missing data points, clean data sets, split train set, test set. It's a semi-process, another application machine learning. Train a model, choose an appropriate model. In this case, in genomic data, I use models based in sequence model to analyze sequence. It's a different model. A train data set to train this model. Evaluate this model. It's important to say. Text accuracy, precision, loss, and not evaluate your model depends on your analysis. I like so much to show this perception because I use neural networks in my analysis. Deep learning is it's a big topic to work with genomics because we need many data. We need inputs, your inputs. It's a simple perception, but in sequence analysis, this architecture is different. It's not a simple architecture. We need inputs, weights, calculus of weight and sum, output, step functions. Step functions depend of your model, okay? And we need results are passing other neurons. It's a simple neuron, it's a perception. But in an analysis, we need a special type of neural network called recurrent neural network. In other talk, I'd like to talk about specific recurrent neural network and with an application. In this talk, I will choose only concept, but in another talk, we need to practical about recurrent neural networks. In recurrent neural networks, we have a variance, so it's called LSTM. But first, let's understand what is recurrent neural networks. Recurrent neural networks, or RNN, is a type of neural network which the output from the previous step it's fed into current step. When it's recurrent network are a memory. It's a memory in this neural network. And simple neural network, you don't need memory this past, but the recurrent neural network we have memory. But in recurrent neural network, we have a specific problem called vanish gradient problem. Not a talk we can discuss about vanish gradient problem. Vanish gradient problem, it's a problem in recurrent neural network, but in LSTM solve this problem. It's in a specific type of recurrent neural network. In other talk, we can discuss about this topic. But let's understand this again. In this figure, we have a representation about recurrent neural network. You can realize in this topic, and the next neuron use the previous result. Memory of the past. Recurrent networks work this form, with the past, with a memory. Some other types of neural network don't work this form. We lost the past, but in recurrent neural networks, we have a memory to use and analyze sequence, because in sequence, you need to identify the past, in some cases, the next. I use other type of approach. For example, LSTM. And let's explain again. Recurrent neural network is a type of neural network in which the output and this first representation x0, we have a result, the output. The next layer, the next x1, we use the previous steps and the current steps, and many epochs. Okay, we have a first, we have a result. The next layer use the output for the previous steps. this representation is good because I looking for a good GIF to represent better these two types of neural networks. X0, X0, X1, X2 is a time. Current time we use the output from the previous step to use in this epoch. And the neural, this type of neural network is very powerful to work with its sequence. But in a specific type, the recurrent network in this form, using the output from the previous step, we can problem about we lost the memory In this case, we use other approach called LSTM. Another talk we need to discuss about LSTM in the a protocol approach this time. Thank you so much to watch this talk. It's my Twitter, and Jéssica Costa Dev is my LinkedIn. We can talk about this topic. We can discuss this topic. I work with this research. In my case, I work with diseases and drug targets. I work with proteins. I don't work exactly with DNA, but in proteins. But interesting, can talk to me for through Twitter or LinkedIn. Thank you so much.
It's so weird to have a meetup online because then you don't hear the clap. So I'm here to give you the clapping that you deserve. So, folks, if you have questions, Jéssica, of course, just shared her contact. And again, this was recorded, so you can still use this link and watch it over. And hopefully, we're going to have her here again to give more talks. What do you say,
People
Jéssica?
interested in this topic can talk to me because I want research to work with me in this topic.
Excellent.
There's opportunity interested to work with me.
Brilliant. Thank you very much for doing this on your Saturday, Jéssica.
Thank you.
Thank you, and I'll see you soon.
Bye.
Bye.
Bye.